PCR in agricultural sciences and environment. As I told you that PCR can not only be used to detect infections that are caused by microbes in humans, but it can also be used to detect different infections or pathogens that cause infection in animals as well as some agronomically important crops or in general plants. So microbes, they can cause different infections in animals, like in birds, uh, avian influenza, it is very common, or some bacterial infections that is caused in economically significant animals. Similar may be the case with plants, that different viral pathogens, bacterial pathogens, or even fungus, it can deteriorate the quality of the crops. So this PCR technique, it has also been used to detect different causal agents of animal and plants as well. In addition to that, if we want to enhance the productivity of the crops or to develop resistance against insects, for example, we can target or clone that particular gene that can enhance the crop efficiency in a particular environment, or it can mediate the plant tolerance against certain insect attack. So what we can do, we can transform the protoplast of the plants by using some specific technique that we will discuss later on when we will going to study that what are the applications of gene manipulation. लेकिन इस टाइम पे इतना ही कहना काफ़ी होगा कि डिफरेंट जीन्स को ट्रांसफॉर्म किया जा सकता है, चाहे वो प्लांट्स हैं या एनिमल्स हैं, so that they will show desirable expression that can be ultimately will benefit the plants as well as humanity. So in agricultural practices, a specific gene, it can be transformed to the host organisms. इसके अलावा हमारे जो फूड प्रोडक्ट्स आते हैं, लाइक फ्रेश एग्रीकल्चरल प्रोड्यूस है या प्लांट्स हैं, तो बाज़ दफ़ा दे may be infected with pathogens, like फ्रेश एग्रीकल्चर प्रोड्यूस हैं, क्यूकम्बर हैं या टोमेटोज़ हैं या कुछ और तरह के सैलड्स हैं, लीफी वेजिटेबल्स हैं जो कि हम यूज़ करते हैं, तो ड्यूरिंग हार्वेस्टिंग और प्रोसेसिंग they can be contaminated with some deadly microorganisms, like कुछ strain जो E. coli की हैं, they may be very toxic, and some specific genes, they can be targeted for the detection, like enterohemorrhagic E. coli. Some genes can be targeted that produce Shiga toxin that is lethal to the humans, like Shiga toxin gene 1, which is commonly called as SLT1 or SLT2, and they can be targeted, and certain strains of Escherichia coli that may be harmful, like E. coli O157H7. This is one of the example of a pathogenic strain that can cause infection to humans due to foodborne nature. So foodborne diseases, they can be prevented by detecting the pathogens, and here again this application, that is PCR, it can be used. Nowadays, different studies have been conducted that showed the presence of different pathogenic microbes and their detection by using PCR. इसके अलावा हमारा environment है जिसमें हम रह रहे हैं, तो उसमें भी different harmful agents जो हैं वो enter हो रहे हैं in the form of pollution, in the form of organic materials, some synthetic compounds are there, और इन synthetic compounds की वजह से क्या है कि इन्वायरमेंट की पोल्यूशन हो रही है, और इस पोल्यूशन की वजह से जो इन्वायरमेंट की क्वालिटी है वो डिग्रेड हो गई है। बहुत सारे हेल्थ इश्यूज जो हैं वो आ रहे हैं, लाइक सम एजेंट्स दे मे बी कैंसर कॉजिंग or carcinogenic. And here we can use certain microbes to degrade them, and even modified microbes, which we can call it as the genetically modified microbes. As I told you that genetically modified crops can be used, and here the genetically modified microbes, they can be produced by introducing specific target genes. And here again the PCR, it can provide the specific target gene so that a particular microbe, it can degrade some pollutant in the environment. So this technique, it can also be used not only in the field of agriculture to detect different diseases or to transform the organism, but to introduce our gene of choice in the microbes so that they can clean the environment for us.